Okay, so we're going to be starting our unit on meiosis. So we've talked a lot about chromosomes, and meiosis is the separation of chromosomes to make gametes. So it's a vital aspect of sexual reproduction that produces gametes. And remember, gametes is just a fancy word for sperm and ovum. And remember, you get half your chromosomes from mom and half from dad. So to do that, the first thing they have to do is make those haploid gametes. So meiosis is a process of producing 4 haploid nuclei from a single diploid cell. And remember, diploid means that you have 2 versions of each chromosome, and haploid means that you only have 1. And maybe you're already detecting a little bit of a problem that diploid, as I said, we only have 2 versions of each chromosome, but we need to somehow get to 4. And to do that, what has to happen before meiosis can even take place is that the chromosomes have to replicate. So if I have, say, these 3 pairs of homologous chromosomes, each one of these is going to have to replicate. So that's going to happen for this chromosome 1. And when they replicate, they are called sister chromatids. So sister chromatids are different from homologous in that they are genetically identical. And these sister chromatids are attached to one another by something called a centromere. It's kind of in the center, so where it gets its name from. And this is going to happen for every pair of chromosomes in the cell. All of them have to be replicated because remember, I need those 4 copies to make those 4 haploid nuclei. And again, each sister chromatid pair is going to come with its own centromere. So before meiosis can even begin, the chromosomes have to be replicated, and this happens during the S phase of interphase. And again, they create these sister chromatids, which are genetically identical. So they were replicated, so it's just a copy. And again, they're joined at the centromere. And so after this, now you have the 4 copies of each chromosome that you need to actually separate them during meiosis. So again, the DNA in a sister chromatid is identical. So this is different than a homologous chromosome. Remember that homologous chromosomes have the same genes, but they might have different alleles. So sister chromatid is not like homologous. They are identical. Homologous, same genes, maybe different alleles. And I think on your notes there isn't a space for that. So how I would summarize that, I would just say sister chromatid equals identical, homologous equals different alleles. That's how you remember that. So know how to distinguish between those two because they are different. Okay. So by the end of meiosis, each chromosome is going to separate into like these 4 different haploid cells, and it's going to be random how these end up. So this is how we end up with more genetic diversity in the ending animal. Okay, so now we're going to talk about the first stage of meiosis, or the stages. So meiosis is made up of 2 different cell divisions, and they're called meiosis 1. On this side, this is the first 4 stages here, and meiosis 2. So on your sheet, you have a picture of this, so you can go ahead and circle it. And for these first 4, that first cell division is meiosis 1, and the second one is meiosis 2. Okay, so just label them like that. So meiosis 1 separates the homologous chromosomes, while meiosis 2 is separating the sister chromatids, which produces those 4 haploid cells. So in meiosis 1, the action is all about the homologous chromosomes being separated, and in meiosis 2 is all about the sister chromatids being separated. Okay, so know that. So, okay, for your warm-ups, at some point you will be asked to draw these from scratch, so do you know how to do that? Okay, so what I wanted you guys to start doing is to know the names of the stages and their order. So I always remember PMAT. So meiosis 1 has this prophase 1, metaphase 1, anaphase 1, telophase 1, so you can use PMAT to remember the order. And you will notice that meiosis 2 is just a repeat of meiosis 1. It's the same thing, same order, same stages, just with a 2 on it. So again, remember in meiosis 1, what is being separated are the homologous chromosomes, and in meiosis 2, it's the sister chromatids that are being separated. So let's look at these one at a time. So for prophase 1, this is the first stage of meiosis. This is when the chromosomes initially condense and first become visible. You can't really see them before that. In pictures of prophase 1, you might have just like like strings, like a bunch of like a pile of thread where they're not really visible. So that would be your prophase 1. Also in prophase 1, the nucleolus breaks down and you start to see the formation of the spindle fibers coming from those centrioles. And what they're going to do is they're going to run around and attach to those homologous chromosomes. So in prophase 1, the other thing that happens is these homologous chromosomes start to pair up. And again, in prophase 1, a super important thing happens, the crossovers between those homologous chromosomes. So we'll talk about that more in depth in the third video. So just know that for prophase 1, what's happening is that these homologous chromosomes are swapping sections. So they're just trading parts. So this homologous chromosome 1 on the inside and this one here, they're going to swap sections. So they'll literally reach out and trade sections. So now you have this sort of unique chromosome that's different from the others. And this happens for every chromosome pair, every homologous pair. They're going to trade parts. So it gives you these unique chromosomes that didn't exist before, and that gives you even more genetic variety. Okay, so put a star on this on your sheet or just in your notes. Know that the crossing over happens in prophase 1. Okay, so after prophase is metaphase, and I always remember metaphase by middle. So this is when the chromosomes line up in the middle, and again, they're being pulled around by those spindle fibers. And the orientation in metaphase is random. So right now, the darker homologous chromosomes are on the bottom, but it could go either way. Like maybe on this one, the darker chromosomes are on top, and for chromosome 1, they're on the bottom, or they could easily be flipped. So that random orientation, it's going to come into play when we start separating these guys. And again, that's another way that we can get genetic diversity. So after they're oriented, however they're oriented, the spindle fibers connect to something called the kinetochore. So this is a separate protein than the centromere. So the kinetochore is what the spindle fibers attach to. The centromere here is what holds the sister chromatids together. So kinetochore attaches to spindle fibers, and that's how we're going to move these chromosomes around. So again, in metaphase, they line up in the middle. And in anaphase is when they get pulled apart. And I remember anaphase by like a Wade, starts with A. So in anaphase, the spindle fibers contract. It's going to pull those homologous chromosomes apart. Okay? So anaphase 1 is separating the homologous chromosomes, because meiosis 1 is all about homologous chromosomes. So in telophase, that's when you can start to see the new cells forming. So the nuclear envelope is going to reform. You're going to get a new membrane. So this here, before it's fully formed, is called a cleavage furrow. And you can always tell telophase because the chromosomes are on the top and bottom of the corner, and you start to see the separation of the cell. And you'll know that it's telophase 1 because you can see 2 cells that are visible. And so you have these 2 distinct haploid cells now. So we're going to pause there before we move on to meiosis 2.